Hey guys, welcome back to another episode of my channel. My name is Keika. I go over textbooks chapter by chapter, section by section. This video we'll be starting Unit 2, The Cell. Chapter 6, The Tour of the Cell. And Chapter 6 consists of, eight key concepts. Starting with 6.1, biologists use microscopes and biochemistry to study cells. 6.2, eukaryotic cells have internal membranes that compartmentalize their functions. 6.3, the eukaryotic cell's genetic instructions are housed in the nucleus and carried out by ribosomes. 6.4, the endomembrane system regulates protein traffic and performs metabolic functions. 6.5, mitochondria and chloroplasts change energy from one form to another. 6.6, the cytoskeleton is a network of fibers that organizes structures and activities in the cell. 6.7, extracellular components and connections between cells help coordinate cellular activities. And 6.8, a cell is greater than the sum of its parts. And learning objectives. Identify how common techniques of microscopy and biochemistry are used in cell biology. Compare and contrast prokaryotic and eukaryotic cells and animal and plant cells. Describe the structure of the nucleus, chromosomes, and ribosomes, and explain their functions. Identify the parts of the endomembrane system and describe their roles in the cell. Compare and contrast the structures and functions of mitochondria and chloroplasts. Describe the subunits and structures of the three types of cytoskeletal fibers and list their functions. Compare and contrast the extracellular components of plant and animal cells and the cell functions of plant and animal cells. And lastly, explain how the parts of a cell work together to enhance cellular function. Okay? So let's get started. Concept 6.1. Biologists use microscopes and biochemistry to study cells. How can cell biologists investigate the inner workings of a cell, usually too small to be seen by the unaided eye? Before we tour the cell, it will be helpful to learn how cells are studied. Microscopy or microscopy. The development of instruments that extend the human senses allowed the discovery and early study of cells. Microscopes were invented in 1590 and further refined during the 1600s. Cell walls were first seen on dead cells of oak bark by Robert Hooke in 1665 and living cells by Antony van Leeuwenhoek a few years later. The microscopes first used by Renaissance scientists, as well as the microscopes you are likely to use in the laboratory, are all light microscopes. In the light microscope, LM, visible light is passed through the specimen and then through the glass lenses. The lenses bend the light in such a way that the image of a specimen is magnified as it is projected into the eye or into the camera. Three important parameters in microscopy are magnification, resolution, and contrast. Magnification is the ratio of an object's image size to its real size. Light microscopes can magnify effectively to about 1,000 times the actual size of the specimen. At greater magnifications, additional details cannot be seen clearly. Resolution is the measure of the clarity of the image. It is the minimum distance between 2 points. It is the minimum distance 2 points can be separated and still be distinguished as separate points. For example, what appears to the unaided eye as one star in the sky may be resolved as twin stars with a telescope, which has a higher resolving ability than the eye. Similarly, using standard techniques, the light microscope cannot resolve detail finer than about 0.2 micrometers, or 200 nanometers, regardless of the magnification. The third parameter, contrast, is the difference in brightness between the light and dark areas of an image. Methods for enhancing contrast include staining or labeling cell components to stand out visually. Figure 6.3 shows some of the different types of microscopy. Study this figure as you read this section. Okay. We're going to skip that. This is 6.3, the one that they just mentioned. We have light microscopy, this is what you can see with light microscopy, or microscopy. And this is what you can see with electron microscopy. Until recently, the resolution barrier prevented cell biologists from using standard light microscopy when studying organelles. Until recently, the resolution barrier prevented cell biologists from using standard light microscopy when studying organelles, the membrane-enclosed structures within eukaryotic cells. To see these structures in any detail required the development of a new instrument. In the 1950s, the electron microscope was introduced to biology. Rather than focusing on light, the electron microscope focuses a beam of electrons through the specimen or onto its surface. Resolution is inversely related to the wavelength of light, or electrons. A microscope uses for imaging, and electron beams have much shorter wavelengths than visible light. Modern electron microscopes can theoretically achieve a resolution of about 0.002 nanometers, though in practice they usually cannot resolve structures smaller than about 2 nanometers across. Still, this is a 100-fold improvement over the standard light microscope. The scanning electron microscope is especially useful for detailed study of the topography of a specimen. The electron beam scans the surface of a sample, usually coated with a thin film of gold. The beam excites electrons on the surface, and these secondary electrons are detected by a device that translates the pattern of electrons into an electronic signal sent to a video screen. The result is an image of the specimen's surface that appears three-dimensional. The transmission electron microscope is used to study the internal structure of cells. The TEM aims an electron beam through a very thin section of the specimen, such as a light microscope aims light through a sample on a slide. For the TEM transmission electron microscope, the specimen has been stained with atoms of heavy metals, which attach to certain cellular structures, thus enhancing the electron density of some parts of the cell more than others. The electrons passing through the specimen are scattered more in the denser regions, so fewer are transmitted. The image displays the pattern of transmitted electrons. Instead of using glass lenses, both scanning electron microscopes and transmission electron microscopes use electromagnets as lenses to bend the paths of the electrons, ultimately focusing the image onto a monitor for viewing. Electron microscopes have revealed many subcellular structures that were impossible to resolve with the light microscope, but the light microscope offers advantages, especially in studying living cells. A disadvantage of electron microscopy is that the methods customarily used to prepare the specimen kills the cell and can introduce artifacts. Structural features can introduce artifacts, structural features seen in micrographs that do not exist in the living cell. In the past several decades, light microscopy has been revitalized by major technical advances. Labeling individual cellular molecules or structures with fluorescent markers has made it possible to see such structures with increasing detail. In addition, both confocal and deconvolution microscopy have produced sharper images of three-dimensional tissues and cells. Finally, a group of new techniques and labeling molecules developed in recent years, called super-resolution microscopy, have allowed researchers to break the resolution barrier and distinguish subcellular structures as small as 10 to 20 nanometers across. A recently developed new type of transmission electron microscopy called cyroelectron microscopy, CyroEM, allows specimens to be preserved at extremely low temperatures. This avoids the use of preservatives, allowing visualization of structures in their cellular environment. This method is increasingly used to complement X-ray crystallography in revealing protein complexes and subcellular structures like ribosomes, described later. Cyroelectron microscopy has even been used to resolve some individual proteins. The Nobel Prize for Chemistry was awarded in 2017 to developers of this valuable technique. Microscopes are the most important tools of cytology, the study of cell structure. Understanding the function of each structure, however, requires the integration of cytology and biochemistry, the study of chemical processes or metabolism of the cells. Okay, so microscopy. In my notes, I wrote, the development of tools such as the microscope allowed for the discovery and early study of cells. Microscope invented in 1590, refined during the 1600s. Light microscope were first used by Renaissance scientists. The light microscope is when visible light is passed through the specimen and then through a glass lens. Lenses refract, bend the light, so the image is magnified. Important factors for microscopy. Magnification. Ratio of an object's image size to its real size. Resolution. Measure of the clarity of the image. The distance between 2 points can be separated and still be distinguished as 2 points. Contrast. The difference in brightness between the light and dark areas of an image. Staining enhances contrast. Important developments to the standard light microscopy allowed for the study of organelles. So we have the electron microscope, which focuses beams of electrons through the specimen or onto its surface. Shorter wavelengths equals higher resolution. Scanning electron microscope, used for detailed study of the topography of a specimen. Uses thin film of gold over specimen to translate a three-dimensional image. Transmission electron microscope. Studies the internal structure of cells via staining of the specimen with heavy metals to enhance electron density of some parts of the cell more than others. Electrons passing through scatter more in denser areas, and the area is a pattern of transmitted electrons. Scanning electron microscopy and transmission electron microscopy uses electromagnets as lenses to bend the path of electrons for viewing. The light microscope offers the advantage of observing living cells, while electron microscopy kills the cells. Light microscopy has gone developments over the past few decades. Fluorescent markers allows us to see the structures with increasing detail. Confocal and deconvolution microscopy have produced sharper images of 3D cells, three-dimensional tissues and cells. And super-resolution microscopy allowed researchers to break through the resolution barrier and see structures as small as 10 to 20 nanometers across. A new type of transmission electron microscopy, called cyro-electron microscopy, allows for specimens to be preserved at extremely low temperatures. No preservatives needed, and complements X-ray crystallography, which reveals protein complexes and subcellular structures like ribosome. Microscopes are important for cytology, study of cell structure, and must be complemented with biochemistry, the study of metabolism in cells. Okay. Cell fractionation. A useful technique for studying cell structure and function is cell fractionation, which takes cells apart and separates major organelles and other subcellular structures from one another. The piece of equipment that is used for this task is the centrifuge, which spins the test tubes holding mixtures of disrupted cells at a series of increasing speeds, a process called differential centrifugation. At each speed, the resulting force causes a subset of cell components to settle to the bottom of the tube, forming a pellet. At lower speeds, the pellet consists of larger components, and at higher speeds, results in a pellet with smaller components. Okay, so I'm just gonna draw for you what a centrifuge looks like. So it's like a circle here. You got a test tube here. You got a test tube on the opposite side. And it's like let me just get rid of this test tube. Okay, and then it spins around really, really fast. So it causes the particles to go to the bottom. Think of like, actually, I don't know what you could think of, but this is essentially the example that I thought of. This is what I remember from my biology class. So this spins around. I don't know if you can see that. This spins around, and the force of the spinning in one direction, depending on how fast it goes, causes the heavier aspects within the test tube to fall down to the bottom. And then what you get is a little pellet. This is the pellet at the end. And then the rest up here is clear water or clear liquid. And then the solids go to the bottom. Okay? This is also an example of a centrifuge. I guess this makes sense as well. So as you can see, the pellets... Oh, let me go back. So this is what it looks like before, and this is what it looks like when it's done. Cell fractionation enables researchers to prepare specific cell components in bulk and identify their functions, a task not usually possible with intact cells. For example, on one of the cell fractions, biochemical tests showed the presence of enzymes involved in cellular respiration, while electron microscopy revealed large numbers of organelles called mitochondria. Together, these data help biologists determine that mitochondria are the sites of cellular respiration. Biochemistry and cytology thus complement each other in correlating cell function with structure. Okay. And for my notes, cell fractionation. It takes cells apart and separates major organelles and other subcellular structures from one another for studying cell structure and function. It involves a centrifuge, which spins test tubes holding mixtures of disrupted cells at a series of increasing speeds, a process called differential centrifugation. At each speed results in a subset of cell components to settle to the bottom of the tube, forming a pellet. Low speed equals larger components in the pellet. High speed results in smaller components in the pellet. Cell fractionation allows researchers to prep cell components in bulk and identify their functions. Example: biochemical tests found enzymes involved in cellular respiration, while electron microscopy revealed large numbers of mitochondria. Concluded that mitochondria are cells of cellular respiration. Okay. So that's it for concept 6.1. Next video, we'll be going over 6.2. Eukaryotic cells have internal membranes that compartmentalize their functions. Okay? So that's it for today, and I'll see you guys next time. Have a good one. Take care.